30 to 50 frames, depending on how bright the beam was on that day. You know, it's either a 0.2 second collection or a 0.3 second collection. Sorry, framing rates. And then we're exposing for about 10 seconds. And so each 0.3 seconds, you have an image or a SAXS profile. And somewhere in here, radiation damage may occur. And it's impossible to predict beforehand when that'll occur. So the advantage of collecting this way is that you get to maximize your signal to noise without radiation. The disadvantage is that you have to process all this stuff, and it can be painful, and deciding when you start getting radiation damage isn't trivial, and there aren't very great programs to do that. We have some issues still with our system, though I think a lot of them have gone away, although you'll still see them, and depending on the run, and again, especially during COVID times, you know, we basically had 2 people who were allowed on site to do all the work that we did over the last 3 months. So, you know, we definitely didn't hit perfection for everything. But some data sets went perfect, and you got perfect. So typically you'll send us a buffer before your samples and a buffer after your samples, and 3 concentrations in between. That's very frequent. That's what we've asked for. By subtracting the buffer before your sample, you get 1 scattering curve. By subtracting the buffer after your sample, you get a 2nd curve. In an ideal world, those guys would be perfect. They'd be a perfect match. But yeah, sometimes that's not true, and you have to decide which is true, which is correct, or if you are going to hedge bets and take an average. So sometimes you'll know in a particular set that we'll get high Q buffer subtraction errors. One thing to note is that this is a log scale here on the intensity. So it's really small differences. It appears large on the log scale that, out here, but it's actually very small differences in intensity out in the high Q. So, you know, you could take an average in this case if you didn't know which one was correct. That would be a safer bet. We have had bubbles in the past. So here's like this is when we're using metal needles instead of 3D printed needles of plastic. So the cells look different. But you know, we had bubbles, so you can check through your samples for bubbles. So please do that. Generally, we provide a set of images for each sample while it's being collected. So there's like a sample image directory where you can check through and look through it. And again, I'm focusing on the tough stuff, right? So things to look out for. Also, sometimes you'll see a small q variation in the very small q. In this case, it's obvious which one is correct. It's the black curve. It doesn't have a nonlinear sort of behavior. The safest bet, of course, would be to truncate the data at, you know, 0.1 something, 0.01 something, and take the average in between if you were really concerned. But you know, these are small differences, and you know, you're going to have to plow through them. So you know, we've been pretty successful. You'll get in your package a set of average images. So this is like going through someone's plate, and they're comparing the first buffer and the second buffer subtraction. And also you get a sense of the radiation damage. So if you average the first 5, those are the dashed curves. And if you average the all of the things merged together, those are the solid lines. So you get a sense of how you know, the issues of your data collection by scanning through those. And you see sometimes you get the high Q problems in certain ones, and sometimes you see small Q problems in certain ones as well. But hopefully for you, you got good agreement throughout. There are varying philosophies on how to process data. If you really want to see like perfection, there's a paper that's titled 2017 Publications. This is a pretty aspirational paper. They provide 4 tables that they, that are useful. If you were to truly fill these out for every sample, basically you fill up a whole paper for each sample. But it's good to read through and to see like kind of an ideal case of how you would analyze the data and things to look out for and work for. These are examples. In that paper, they show you the tables that they'd put out for each of these cases. So it's good to have. Sometimes you'll run into a reviewer who is of this purist school of thought and think that a paper for every sample, you need to have this. In that aspirational paper that I was citing here, we did insert that, these guidelines are not intended to restrict publication, but just as guidance, okay? And also that, when you're collecting data in high throughput screening, if you're comparing constructs or metabolites, you're not going to hit all those things. But there's still some really critical quality control parameters that you really want to make sure. You want to check your RGs, which you can extract, your race duration, several different ways. That's quality control. Your flexibilities and your masses, make sure you probably have an idea of what the mass should be. So that mass should correspond to something that makes sense to you. You can, if you get something that tells you it's flexible from the SAXS measurements, you should check with secondary structure prediction, obviously, and see if flexibility is there. The two should correlate. And your mass should be about 10% of what you expect it to be. So those are like quality controls that you can check back on to make sure your samples are good. All right, and I'm approaching that mark there. So I'm just quickly going to go through. I'll try to get that paper in the just the chat. So let's see. You'll get a folder from us and a login from us that kind of looks like this. You've got that folder that has these images, so A3, A4, they were low concentrations. So you got like a big idea of, things that worked out and things that didn't work out, buffer 1s and buffer 2s. When you started to have radiation damages, for example, you should be able to scan through and use that sort of AvGraphs picture folder for that. Sample cell pics are here, so you saw a bubble there in this particular set. But I'm going through, and sometimes you have a bubble, sometimes you don't. Things run well. Let's see. So so let's see. Let's go to. So you got all your data. So you got subtracted folder and unsubtracted folder. In the subtracted folder, we've subtracted buffer 1 and buffer 2 from your sample. In the unsubtracted buffer, you have all your files. The circularly integrated images for all for buffers and samples, which you can manually subtract from one another. If you look in the subtracted folder, this is what I prefer to work with most. You'll notice, for example, that A1 is missing and A5 is missing. That's because they were buffers. And you've got your samples here. You can go to one of your folders here, let's say picking D4. And there you've got your buffer 1 subtraction, it would have been D5, or sorry, D1 subtracted from D4, that would have been buffer 1, and then D5 subtracted from D4, that would have been buffer 2, and then the average of these two in the average folder. All right, so that's how that works. And then if you go into buffer 1, you can see all the data, the 33 frames. So this was 10-second exposure, framed at 0.3 seconds, all 33 frames, all right? And now you gotta go through a program that will help you figure out when radiation damage occurs. Now, I'm going to show you 3 ways of doing that. One way is to log into our system, and that's not the most convenient way because you have to log into our system. But and unfortunately, this is the web version isn't working very well right now. So that's one of the reasons I'm giving you 3. We're going to fix the web version today. And I'll demo that for sure. But if it's not working and you're super hot to get this going, we've got a command line version, which you can copy. If you log into our system and copy it, you do, this is all Python code. Anyway, so you can, so it's this one, regions and num files, and then *1.dats. And so what it does is it separates the data to 3 regions: a Guinier region, a Poirot region, a wide Q region. And you can start adding frames as you go along. You can zoom into the Guinier region, and you can kind of start adding frames in. And you can see as I'm adding frames here that I'm starting to get some radiation damage. So, I can add up to a certain point, somewhere around here, maximize my signal to noise. It's about 4 frames in this region, in the Guinier. I can extend the Guinier region too by, right now you see that the Guinier region ends at around 0.45. I can extend it with this other bar, and this is true of the web tool as well. So you've got basically 5 parameters to mess with. But anyway, you're trying to maximize signal to noise. So here it's 4. Because radiation damage affects the small Q region the most often, unless you got a metal center. Sometimes you have a metal center and it's coating a bunch of amino acids. That might get damaged first, and the amino acids might pop off and start unfolding in that specific area. So you might see that your Guinier region stays fine because it doesn't change the overall RG, but it's changing sort of the fold. And sometimes different parts will get damaged first. So here I can do about 9 frames before I really get into like a step function here, which I do not want. I do not want a step function. If you introduce a step function into something that you're going to Fourier transform, you're going to end up in trouble. So you want a real smooth transition between your sets. So around 7 in this area. And again, I could probably increase the Guinier region. And then I can hit home here and, add a few curves onto the wide Q. And generally the wide Q area, you're probably pretty good all the way through. And again, this is where the ability to adjust where the regions end and start come in. So I could probably bring this region in a little bit until it deviates. And so there's 3 curves showing up here. There's the original first frame, which is the gray. There's all of the exposures, which is red. And then there's the black curve, the working curve. All right, so then I've got, now I got a final curve that I'm happy with, and I can hit home here. And in this particular version of this frame slicing, the frame is saved as defined by, as an output here. So I've got a curve that looks like that, and I can plot it on its own. That's the curve. Going to the web tools. Let's see. And again, this wasn't working really well for me today. And I think I'm just having a data log. So sometimes the data is not being passed well. So at our website, SIBYLS.lbl.gov, you hit the SAXS, you hit this frame slice. You can go in now into your directories that you've downloaded from our Beamline. and you can start pulling data in. And I would pull all the data in except for right now, I get some slight errors. So I'm pulling in just 10 of the frames right now, but you should be able to pull in, you should be able to drag and drop the whole folder, like buffer 1, buffer 2. Okay, so it looks a little different, but it's still the same idea. You got 3 toggle marks for Guinier, Porod, and YQ, and then you can adjust where the regions end and start. Again, this is the way to go here. In this particular region, there isn't a lot of radiation damage, but you can click into the Guinier region here by hitting Guinier. You can move the scroll bars around. There's a lot of little tools here for helping you out. You can add frames. And again, there's 3 codes here you can't see very well, but it's blue is the original, orange is all frames integrated together, and the darker one is the current set of frames. Here, pretty much you can use all frames. And of course, I'm just showing the first 10 because I had some download issues. But yeah, certainly play with that as well. And then finally in scatter, let's see. Scatter's got a tool in the subtract mode. You dump in all your, from the unsubtracted data, you dump in all of your sample data, all of your buffer data. It plots the samples and buffers, and then it kind of comes up with a metric of similarity between them. And so as long as this is all pretty flat, you can, hit subtract all, or you can limit this to, 12 frames or something like that, and then hit subtract all. So that's the third way of doing all of this. Let's see, I think, what did I want to show? Oh yeah, someone asked about, let's see, where did I put that? About concentration dependence. So you collect 3 concentrations. I know I put it in here somewhere. Here it is. So you collect multiple concentrations. The reason you do that is because your sample may be attracting itself or repelling itself. And I'm already at 11:30. I promised Mikkel I'm going to let go. You want to make a correction for the concentration dependence. And basically, here's a concentration dependent series right here. You can see that the small q is changing as a function of concentration. You got to scale the data to get an accurate view of concentration. And then for each q point here, you're going to want to create a graph like this, where what I'm doing is taking this q value and I'm plotting the intensity at that q value over here, and the concentration. And the more concentrated this sample is, the more down it is in the small q region, and that's because it's repelling itself. So at this q point in the high q, I got this green dot, that's the intensity there. Purple at that q value, the sort of middle high concentration, middle low concentration, and then the lowest concentration. And then I want to extrapolate what the value would be at 0 concentration. And that would be a black curve, and I do that for each q point. So that's physically what you're doing. I learned recently that Primus has got a tool for that. So if you download Primus, the Qt version, and you put in your concentration series here, you have to add in the concentration that you expect it to be, and you want to make sure that's done accurately. And the best way to do that is to scale the data and see how much you have to scale the data. In other words, it's concentration dependent. Once you've entered the concentrations here, you hit the extrapolate button, and it gives you a extrapolated small q. Oh, sorry, I'm not showing them. The 3 curves here. And so these are the 3 concentration sets. I entered the concentration series, and then I hit extrapolate, and it gave me what it thinks is the concentration dependence. So use that tool if you don't want to write your own script or use an Excel profile. I think with that, I'll stop. I'll see if there's any questions. And the idea is now to pass it on to Mikkel for him to do the same for SEC-SAXS. Hopefully you guys are still out there. Someone's asking about whether you scale to I0 or to the highest concentration. What you actually want to do for scaling is when you hit scale here, let's see. And again, I'm doing this in Primus. You can do this in Scatter, but you can hit this scale button here. And you can see that they don't all scale in a right way. They're scaling to the wrong region, to the I0. What you really want to do is scale to somewhere in here where all curves are the same. So here we go from 50 to 100. And then if I look in that area here, I'm not used to, there's a zoom out in here somewhere. Zoom, fit to window. Let's see, so in this region, all the profiles are pretty much the same. Not quite, I haven't quite gotten it, but somewhere around here. That's where you want to scale everything to, where there is no concentration dependence. You want to find the areas where there's no concentration dependence, scale that, and use that. Okay, and someone's asking about recording. I think the recording I think we're recording, and I hope we drop it on the YouTube channel. Yes. All right, with that, and I'm sorry for taking longer, Mikkel, take it away, buddy.
So basically what I'm gonna talk today is just how you deal with the data package that you get from Size-Exclusion, SAXS, moles. Just for those people that never tried, I would just show you what is SAXS moles mean to us. So in our mail-in page, there is just all introduction, how to deal. This is introduction and instruction. And so basically we have a different buffer reservoir that we can choose. Then we have auto samplers and go through the column. And then on the column directly afterwards, we have a splitter that can collect sample for SAXS. It's going straight to the SAXS, or it goes straight to the UV and moles. Multi-angle light splittering gives us basically molecular weight for the same sample and the same buffer in the same time, as we can collect the frames for SAXS as the elutions go after the column. This is very autonomous, so we can really accommodate lots of users. So we can swap automatically between different buffers and equilibrate buffers for different columns. And thanks Daniel. Daniel is not present right now, but Daniel did most of the work during the COVID-19 time for users. So it worked well. You got some good data sets, and I will show you what to do with the package. And Catherine also communicate with you when the samples are ready to look at. So what we have here, how the package looks like, and I'm not so prepared as Greg. He is way better in this, but basically, this is your package. And in a package, you have a bunch of samples. 0, the name of the user. 0 is normally the BSA, that if you want to look at that, it's BSA in your buffer, and also how the signal looks like and it's supposed to look like. for the BSA if you have some doubt about what we did. Then you have PDF files for the moles. You can look at those. You have also raw data for Astra software that is basically bias software that we're using for analyzing the moles data. But you have also this PDF file when the information is about elution and molecular weight of your peak and how much aggregations you have, etc. And then you have also some data files that I will show you later. But let's just give some very basic, simple example of the data what you get. All the time when you look at the data from Size-Exclusion SAXS, you really want to click on the moles data. It's just users don't try to forget that and go straight to the SAXS. So what tell us, this is the profile or PDF file. And it's very slow for some reason. My computer is a problem. Let's just do this again and open the PDF file. Hopefully it's gonna be way better. Okay. Okay, so you have obviously some profiles, light spectrum UV, and you already see there is some huge peak, first peak that is splitted very close to the void. So you have some aggregation problem here, and the peak of probably peak of interest. So that at first, and then you scroll down if you don't want to go all the details about radius of duration. So then you have some information about the molecular weight. And so we look at the molecular weight. So you have about 65 kilodalton, and if it's correlated to your sample, and then you have something close to megadalton, 800 kilodalton, what is close to megadalton. That's probably the most likely the aggregation or something very huge. But this is your peak of interest where you have the molecular weight in daltons. So that information tell you, okay, what oligomerization state I have for my peak of interest, and if it's correlate to what I'm supposed to have. We're using the scatter to analyze our profiles. So let's go from scratch. So this is the top called Size-Exclusion column, SEC. You click on that. This is a scatter 4. And in your package, you already have pre-analyzed and this club 1 is the user, sample 1, and SEC file basically can be dropped directly to, and I have a problem because I'm working outside, the internet is not working well with me. So that will give you a size exclusion profile, and this each dot is a frame that we collect, and each dot corresponds to one scattering profile. And so then you can automatically, it's selecting you some buffers, but if you don't trust automatic selection of buffer, you can go here, and obviously you select the buffer close to the peak before the void, and you hit the update. And that allows you to basically now look at how the profiles look like. And so you see the 2 peaks here, obviously from look at this peak, that's the peak of interest. You need to click on this map, what is the similarity map for each frame to frame. And the most cyan color is more similar. So you obviously don't want to merge this type of these frames here because you see the red is dissimilarity between the frames. And obviously that aggregations are also here, but there are also dissimilarity across the aggregations. So that's what we see in the moles. There is a splitting peak, so there is some large aggregations and some non-specific aggregation in front of the peak. But what I want to say, when you look at this curve and you merge that, you obviously see that this is aggregation when you plot it in the radius of durations. But also molecular weight tell you this is aggregation. So let's focus on more well-defined. So we're gonna click somewhere here. Sometimes with people, you can just go and click in the center of the peak and it's still very cyan colors around it. So the frames are very similar. You give it to name and everything what you need to do afterwards, you just click the merge button. What it's gonna do now for you, it will merge the frames that we selected. And when you go back to analysis button, you will see your merge data appear here. You already have some preview for Kratky plot, your scattering profiles, and total intensity profiles. That was a simple case, and then you can continue with the data processing and analysis. We don't want to go just brief. Just look at the Guinea plot, and you see it's a pretty linear Guinea plot. You can probably don't use those first noisy data, so you can kind of deselect that. But you have the linear Guinea plot indication that you have something aggregation free, and as you see also in your size exclusion, the aggregation being nicely separated from the protein. Okay, so let's go a different case that is much more difficult, and how we're gonna look at that. We clear the samples, clear the buffers, and let's just take user MH1. First, again, as I said before, you look at your size exclusion and moles data. This is right here. And what you already see here, there is some very strong peak for the void. So it's aggregations. And there is some separation of the secondary peak that is probably a sample of interest, but it's super, super close, and it's very transient in understanding of different monomerization state and different aggregations. So I will not even probably bother to look at these data sets, but it will be interesting to see it, how this look like. But it's mix of aggregation and large oligomerization states. So that's what we can say, and already have this pre-analyzed data. Come on, we can do it. Yep. Okay, so this is exactly how this looks like. It's very similar to your moles data, and you see this is somewhere the void is, and this is your protein of interest. Just look at that. You will receive something what is the scattering. Now the question is how much of aggregation, how much different heterogeneity is in the scattering profile. So you have multiple different oligomerization states, but you still can process the data and see how the curve looks like. And but when you do some basic merging of the peak, right? Let's take the back of the peak if we can get some better data sets. We merge that. Okay, we get to the button. My computer is very slow today for some reason. So this is your profile for something what is very heterogeneous. And also when you look at the Guinea plot, it seems hard to define, especially when you look at the Guinea plot, you kind of already can say there is some partial aggregations happening. So in this case, I would be very careful to further analyze the data. But let's get a little room for something in between, what seems doable, but very important for you, because you guys sending us the samples, they are most likely very difficult to do with the cryo-EM or with the crystallography. So that's why all those cases have some aggregations or have some problematic. So this is a sample from the same users, and you see the void is here, and the separation of the void is better in this case. So you have a peak that is well defined. We again don't get to look at the moles and define the molecular weight, and that's this case here, right here. And what you see, the aggregations are here, and the peak is somehow well separated. So now we have a chance to analyze this sample. That's sample number 3 from the user. And just look at the molecular weight. That's helped all the time. It's about 350 kilodalton versus some 600 kilodalton. What is the aggregation type? Larger stuff, and 300 kilodalton seems to be a reasonable number for something what is really big. Okay, and so now we're gonna analyze and merge the peak. Again, we look at the area of interest, and you see the cyan colors are right here. That's will be good enough. And we select, this is the region where the frame's gonna be merged together. And by typing merge, it's gonna merge those frames and give it your, and this is your sample. And it will be interesting to compare the poorly separated sample from better separation of the peak. And that's, you can just type log here, and you already start to see that the aggregations pull the curves up a little bit. So the large aggregations are really responsible for a high intensity for the sample 1, and then sample 3 somehow have the system is well behaved and can be also shown as relatively, we have this first point and we're going to read over it, and you can somehow define a linear region for the Guinea that's tell you the aggregation free state. And 77 is something reasonable for something very large, what is 300 kilodalton and very extended. Anyway, so those are examples how we're using scatter. Some people that I communicated yesterday had a problem with the scatter. There were multiple issues. It could be Java, could be that using the scatter 4 that is not the last release, or you just basically don't like, or your computer don't like the scatter. There are other programs that we can use the same way. I will not show them here, but I just want to mention that is so-called that I personally like a lot. It's more advanced program that we're going to discuss through our workshop in August. And deconvolute the different peaks. If the peaks are very close to each other, we can using the multiple frames and multiple SAXS frames to deconvolute using single value decomposition to deconvolute the different species that are under those two peaks that are very close to each other. So for some reason, my computer is very, very slow, but anyway, so this is a raw written by, and most likely by Jesse and Richard Killian, but that's the program that I will recommend to use in case you're more advanced and you want to use, you don't want to use scatter. I have 10 minutes more. Would probably stop talking if there is some questions. And okay, so there is no question for size-exclusion SAXS, what is good. I like that. I may probably show one more example. The purpose of this 1 hour discussion is not going in data analysis, but how to process the data and understand it. I forget to mention that when let's just take one random sample. Okay, let's just user told I was this March, so it's close. And we get first look at our, oops. Yeah, so again, a difficult example when you have very broad peaks or multiple peaks, but it's well separated from the aggregations. It's a very low signal as well. So I'm not sure if that's gonna work, probably the low concentration. But yeah, let's just take the sample and put it in Tolia 1. Where is Tolia 1? Right here. Okay. Okay, so this is the peak. I now understand why we see such a low signal in the moles and UV, because the sample is not worth to even analyze. The peak is very small, and you probably will see something like this, what I would not considering worth to continue. But that's also tell you, look at the moles first and the UV signal in your report. Let's say look at the TOLIA 2, just for fun. Okay, so you see the peaks are getting a little bit better and bigger. Right? This is already worth to analyze. Still very low concentration. Let's try it if we can see and resolve two curves from this very low concentrations problem. And so here is the sign that probably for this peak. Okay, and I call in my report, call TOLIA, and I call it peak 1. So if you got the peak 1, peak 2, peak 3, it's basically peak 1 is the first peak. And the peak 2 will gonna be basically the second peak. That's a little similarity right here. I could probably use a different buffer region, as you can see already. There is, for such a low concentration, especially in this case, if you're using the buffer region, there could be, in this case, very non-linear. You have a problem to select the buffer, and where did you end up? When you look at the data itself, Sketcher will automatically truncate the data 17 points from the beginning, because when you look at the data, the old data sets, now you understand why you automatically get rid of this chunk of the sample. That's reason why we have such a low concentration, because we have such a low concentration and the buffers are really close to our signal, and we have some fluctuation in the baseline due to our systematic errors, but also due to slightly variation in buffer rates going through the column. That could happen too. But in this case, we know it's a very small, RG is 22, so you just go and cut the data and Sketcher do it for you automatically. You can use the Guinea when you look at all the data, so you will see that there is some problem here. And so you try to find the area when it's linear, and that is this. So you are welcome to cut 25 points in this case, because your protein is very small, 25 kilodalton. This where you got a nice decent Guinea. And don't bother with this first 20 points. So also this very weak signal, when you're merging multiple frames, give you a nice curve. The scattering profile is nice, and in this case, we can say that the peak 1 and peak 2 are very similar, surprisingly. And in this case, you can scale it and look at the differences between peak 1 and peak 2. There are some slightly differences. I would not comment on it right now, but yeah, there is a little difference, but I would say it's very identical. For this low concentration, it's surprisingly identical, even though it's come from the two peaks. Okay, I would say again, with your moles data in your package, you got also some very general report. If you look at your report, for example, this is for the user, that you got the report as well, you got the basic information about your curves. So all this is done for you. You don't need to do that. You can take the final scattering profiles that is in the package. But if you want to reanalyze and choose a different buffer regions, or be careful with the peak that is really your peak of interest, please do it. But you got this SAXS report just informative. You don't want to really take this too serious and just take this is my final results because I said so. You're welcome to try it again on your own. And if there are any questions, raise your hand or come to our workshop that's happened, Greg, you know better, 27, 28?
Yeah.
27, 28. We're going to talk more in details, and we have also very good speakers lined up for Thursday.
All right. With that, maybe, I don't know, Mikhail, you got a couple more minutes. Maybe we take 3 user cases, max, spend 10
Yeah.
minutes. For those of you guys who
Okay.
don't have a specific question, you know, you guys feel free to split. But if there's some user cases out there, maybe 3, whoever hits it first, you can say, I want to see my data analyzed real quick. This folder, this file. Top 3 takers. Sounds like everybody's got an hour.
Yeah.
All right. All right, cool. Well, that makes our life easier. If you guys have other questions, email us. We'll do our best to answer via email as well.
I think we're gonna do this next week as well. So I think if somebody missed something, and we probably gonna take a different examples, and we go probably a little bit more ahead with data analyzing, analysis of size-exclusion data, or finally when you get this final curve, it's all the time the same curve, but it's
Sure.
raining.
Yeah. That's a good point, Mikhail. So we're going to hold this thing again next week, and depending on who shows up, we either go into more detail if it's a repeat of this group, or if there's new groups, we might have to repeat what we did today. So let us know if you're interested. All right.
Okay, guys.
Happy weekend.
Have a nice one. Oh, yeah. Somebody will be there already. We have already one user. Let's sign up. That's good. Okay. See you guys.